Saksha, where every clue begins at level. When there's no forensic serology is located, today you'll be learning about different presumptive and confirmatory tests how to become very clear. So let's start with it. Presumptive test and confirmatory test of coronavirus. First, we learn about blood. The presumptive test of blood. The first one is Casey-Meyer test. So the basic principle of this test is that this is actually based on peroxidase like of hemoglobin. Hemoglobin actually catalyzes the breakdown of hydrogen peroxide and then releases the oxygen, oxidized phenolphthalein to chemical. So this is actually the basic principle of Casimir test. Procedure of Casimir test is that we have to just apply the phenolphthalein onto the suspected stain and then we have to add hydrogen peroxide onto it. The result will be instant pink color. So the phenolphthalein changes the pink color in the presence of blood. Okay, so the significance, why is it significant in forensic science? It is actually quick, sensitive, and also useful at crime scenes. But this test is not specific at all. It's not a confirmatory test. Next presumptive test is accident test. It's actually a historical test. So this uses the same principle of Kaizen test, where phenolphthalein and hydrogen peroxide are used. First, we have to apply the phenolphthalein to the stain, and then we have to add hydrogen peroxide to it. Then a blue-green color appears in the presence of. So this is also actually extremely sensitive, but this lacks reliability. reliability. So this test is also carcinogenic in nature, and because of that, this test is not much used. But second time it was widely used. The confirmatory test of blood is the gamma test. Uh, in this test, actually, formation of chromosomal test. Sorry, in this test, actually, formation of hemochromogen crystals is taking place in the presence of hemoglobin and pyridine. Okay, so hemoglobin is present in the blood and pyridine is present in the Takayama reagent. We have to just add the Takayama reagent to the suspected stain and then slightly heat it. So the result is like pink feathery crystals under microscopic view. So pink feathery type structures will be found when we see through a microscope. Significance is that it can be used for old and degraded stains also. So this is the most important test. The climate test is the most important test, and this is most reliable confirmatory test for blood in forensic science. Now, treatment test. Treatment test actually works by transforming hemoglobin to heme crystals. These heme crystals are also known as periprotopal fiber. So this actually forms in the presence of glacial acetic acid and halide salts. So procedure of this test is that we have to just add the reagent, the treatment reagent, then we have to just add the reagent to the suspected stain, gently heat it, and we have to examine into the microscope. Result is that brownish or so the brownish rhombic crystals are formed in this. Yes, the rhomboid shaped crystals in brown color is formed when we view it under a microscope. The significance of this test is that this is a classical confirmatory test. This is sensitive, but actually it requires more careful heating. So next test is the DNA analysis. We all know that like DNA is available in blood. Actually, it is available in the blood cells known as leukocytes. So we have to extract the DNA from it, then analyze by the process like PCR, STR tagging, or sequencing. So it actually produces a unique genetic profile of the person. So person identification is possible by the blood cell. Uh, the significance of this test is that it is actually most powerful tool in forensic science. Actually, this can be used for individual identification, as I have told you, or even paternity test can be done by this test. And criminal case work is also used for criminal case work. So next fluid is semen. This is also very important fluid in forensic science. The presented test for semen is that acidic acid phosphatase test is also known as AP test. This is presumptive test, and mm, this actually works on a principle that acid phosphatase enzyme is present at a very high level in semen, and actually it hydrolyzes phosphate esters and liberates colored product with the substrate. This is actually the basic principle of this acid phosphatase test. Now, the procedure of this test is that we have to apply alpha naphthalene phosphate to the stain, and then upon it we have to add false blue V reagent. So the result of this test will be purple bright color. Like this is very instant. Like within 30 seconds we will get the results. So if we get the results, we can say that semen can be present in that state. Okay. The significance of this test is that this test is very sensitive. But a drawback of this test is that acid phosphatase is actually not at all specific to semen, right? Like in the previous video, I have told you that acid phosphatase can be present in many other body fluids, including vaginal fluid or even sweat and everything. So yes, in urine also acid phosphatase can be present. So these kind of fluids can give false positive for this test. Next test is Barbieri's test. So the principle of this test is that spermine present in semen actually reacts with the picric acid. Okay, so how can we perform this test? We just have to add the saturated picric acid to semen stains, uh, to semen stain, um, and then observe it under the microscope to view yellow needle-shaped spermine pictrate crystals. So the crystals formed will be yellow in color, and the shape will be almost needle-shaped. Significance is that this test is actually a classical presumptive test, but is now rarely used because it's not so specific, so it's rarely used. So the confirmatory test of semen is that prostate specific antigen test, P30 test. So the principle of this test is PSA is also is a protein. Okay, the PSA specific antigen is actually a protein produced only in the prostate gland. Prostate gland actually produces the semen, right? So this antigen is only present in semen. No other body fluids will have this prostate specific antigen. So result, like how to perform this test, we just have to introduce a strip known as immunochromatographic strip onto the suspected stain. The result will be positive. Okay, the result will be positive reaction confirms presence of semen. If it's negative, then we can say that there is no semen present in that state. What is the significance of this test in forensic serology? Actually, this is a strong confirmatory test even without sperm cells. Yes, some amount like some stains of semen might not contain the sperm cell, right? So even if sperm cell are not there, the semen itself can prove that it is of a male, right? So in in azoospermic cells, males, in azoospermic males, the sperm cells won't be present. So in that type of male also, we can prove that by the presence of semen itself, we can prove that this is actually a seminal state. And this test is actually accepted in both scenarios. DNA profiling. What is the principle of DNA profile? Actually, DNA is extracted from sperm cells uh, or epithelial cells if sperm is absent. As I have told you, in isospermic males, sperm cells might be absent. So, in that case, we can take the epithelial cells. DNA can be extracted from it, and short tandem repeat analysis generates genetic fingerprint of that person. So by knowing the genetic profile of that person, we can get to know about that person, right? So it produces unique DNA profile of that person. Significance of this test is that it actually confirms the identity of semen donor. So this is actually very much critical in sexual abuse cases. Next body fluid is saliva. Saliva, as you see in the pictures, it is actually most important evidence where a cigarette butts or a glass cup is used because the saliva can be present on these things because we use it by our mouth. So the presented test of saliva is the starch iodine test. The principle of this test is saliva contains actually alpha amylase enzyme which breaks down into starch. So normally this starch and the iodine gives blue-black color, right? But if amylase is present in saliva, starch is actually digested by the enzyme. Enzyme means amylase. So iodine cannot form any color. So this principle is actually very easy. Saliva, we know that it contains alpha amylase enzyme, right? Like this alpha amylase enzyme breaks down into starch. Okay, so normally the starch and iodine is actually our reagent which we will be using on this particular test. So when it reacts with this, a blue black color is to be produced. And if blue black color, like if the amylase is present in the saliva, that means if the amylase, which is usually found in the saliva, is present in that stain, that suspected stain, we can say that the amylase reacts with iodine and no color is formed because the amylase actually digests all the starch that is present in the saliva, the reagent. So in this case, no color is formed. The procedure of this test is that we have to just apply the iodine starch solution to the suspected salivary stain. So observe the color reaction. We have to just observe the color. Disappearance or reduction of blue-black color means that there is saliva present in it. That means amylase is present, then saliva is also present. Like as I have told you, this is a presumptive test, so we cannot confirm the presence of saliva just because of the color change of the material. So we have to move on to the confirmatory test to confirm the presence of saliva. Next one is Paribas test. Paribas test is actually widely used nowadays also. So the principle of this test is it uses blue dye star substrate actually and amylase present in saliva actually hydrolyzes starch. So then after it gets hydrolyzed, it actually produces or releases a dye. This is the principle of this test. And how do we have to perform this? We have to just apply the Fariba's tablet or solution onto the suspected stain. Like if it's saliva, then blue color is released. Blue color is shown, right? The significance of this test is that this is more sensitive than starch iodine test. So this is actually very widely used in publicity labs. Next one is radial diffusion method. The principle of this test is that saliva actually simply diffuses through a starch containing gel, right? So amylase activity produces a clear circular zone. Uh, now, how to use it? How to use it actually? Place the sample in gel walls. Just we have to place the sample in the cell walls and incubate it. So the result will be like some amount, some zone of this gel will be cleared. So this zone of clearance indicates that amylase activity is there. That means saliva is present. The zone of clearance is the confirmation, like uh, is the indicator of saliva can be present. Significance of this test is that it's actually useful in semi-quantitative measurement, but this test is actually very time consuming. So this is not much widely used in forensic science. Now the confirmatory test, the RSID test. Yes, RSID salivary test. So the principle of this test is that immunochromatographic test for human saliva, alpha amylase. So and the procedure is that we have to just apply the extract, the salivary extract to RSID test strip. There will be a strip available for this, and we have to just apply the salivary extract onto it. The colored line on the strip actually indicates the case of human origin. Like if a colored line is produced, then okay. The significance of this test is that highly specific for humans. Verification of human saliva specifically, we can use this RSID test. So this is actually very much accepted in forensic practice. Okay. Okay, now the test is DNA profiling has been used in the test. So saliva also contains some ductal epithelial cells like with nuclei. So if there is nuclei, then we know that DNA will be present in that. So DNA can be extracted and profiled by STR analysis, right? So the results will be it produces a unique DNA profile, and the significance is that individual identification is possible in blood samples. Like in cases where cigarette butts or bite marks are present, or food items are present, or glass is present, these tests can be used for the confirmation of what sample is this, whose sample is this by DNA profile, right? Now the next fluid is urine. The most important secondary test of urine is urine nitrate crystal test. So this actually uses a nitric acid. The nitric acid, this test actually works in a principle that the nitric acid actually reacts with the urea present in the urine to produce hexagonal crystals when viewed under microscope. So we just have to add a few drops of nitric acid onto the suspected stain, and then keep it for some amount of time so that it can encourage the crystal formation, and then just view it under a microscope. Most probably this common microscope is preferred actually. So use it and view for the and look for the hexagonal crystal test. If hexagonal crystal tests are present, then we can say that this can be urine. But this test is actually not at all specific at all. Like it cannot prove that this is urine because many other body fluids like saliva, sweat, and tears, which has urea in it, actually produces false positive for this test. So this test isn't enough for proving that this is urine. All right. Now we are moving on to the creatinine test. We just have to add the picric acid onto the stain and then NaOH onto the like suspected sample. And this actually produce a orange red color to the stain. So if orange red color is present, then we can say that creatinine of urine is present in that sample. Creatinine is actually an important element of urine, right? So by this test, we can prove, we can say that, we can detect the presence of urine, right? This is still not a confirmation. We will study about confirmation in the next slide. So, what is the significance of this test? This actually is a quick test for urine detection. As I've told you, this is not a confirmatory test. So, what are the confirmatory tests for urine? Let's get into it. RSID urine test. Like RSID urine test actually detects tan household protein in the urine. So this TAM household protein is actually very specific for the urine, and the procedure of this test is that apply the extract to the RSID test strip, which we have already used in the salivary testing for the like in the confirmatory test. So this actually produces a colored line. So human urine can be detected by this test. So this test is actually very human specific, and this is actually reliable confirmatory test used in forensic case. So next one is microscopic crystal analysis. Urine actually forms distinctive crystals like uric acid, calcium oxide, etc. on drying. So when we test this urine itself under a polarizing microscope, then these kind of crystals can be seen. The figure is given here, so just look onto it. So, what is the significance of this test? This actually is a supportive confirmatory test. So, note like it is not as specific as RSID test, but this can support the other tests, right? Even if we perform the microscopic crystal analysis, we have to perform RSID urine test also. Now, DNA profiling of urine, there is one more test, DNA profiling. So, DNA profiling is actually done on the principle of like urine generally has low DNA, right? Urine has many chance of getting DNA in it. Like even if DNA is like even if cells are present, there might not be nuclei in it. Nuclei in it. If nuclei is not there, DNA might not be there, right? So if epithelial cells are present, DNA can be extracted if there is nuclei in it. So the results of this urine DNA profiling is that it is actually a partial or full DNA profile. Uh, like we can get a partial result or full DNA result from DNA analysis of urine sample because the DNA present in it can be fully developed or partially developed based upon the nuclei present in the cells. So the significance of this test is useful. This test is actually useful when all other tests fail. Okay, this is actually having limited sensitivity but variable in some cases. Okay, now we are going to study about sweat, the presumptive test of sweat, chloride test. The principle of this test is sweat actually contains a high concentration of sodium chloride. Yes, NaCl. So when silver nitrate reacts with this chloride ions, white precipitate of silver chloride is actually formed. So the procedure of this test is we just have to extract the stain into distilled water, like the stain I've actually collected using a filter paper, Workman's filter paper, and then like we can take the stains from the surface or the body itself, and then we have to submerge in a test tube which contain the distilled water, and keep it for almost 2 to 3 minutes so that set can be absorbed by the distilled water. So after it gets absorbed, we have to use it in our test. So this is how we extract this sweat sample into the distilled water, and then we have to just add the silver nitrate solution onto it. We can see white precipitate forms. So this actually darkens on exposure on light. The white precipitate actually darkens on the presence of light. The significance of this test is that this indicates sweat, but it is not a human specific test. Like sweat can be of any other animal, right? So this is not a human specific test. Like similarly, salt can also come from many other sources also. So this test we cannot rely upon this test because salt can also come from many other sources. Next presumptive test is alternative light source. The principle of this test is that sweat stain may fluoresce under certain wavelengths of UV light, which is basically blue light. Okay, how to use it? We just have to use the forensic light source example, 450 to 470 nanometer, to visualize the stains. Okay, the result of this test will be that sweat appears as fluorescent or dark patches depending on the substrate. Okay, so this is actually a percentage test. Like anything can show fluorescence, like different substances can show fluorescence. So this might not like confirm the is I'm sure that this does not confirm the presence of sweat. So the significance of this test is that useful for locating stains which aren't visible to naked eye. This is actually not confirmatory. Confirmatory test for this sweat is that protein marker detection, which has a principle of like sweat has proteins like dermicidin, which is very unique antimicrobial peptide secreted only in sweat glands. Okay, so how to perform this test is that immunoassay or electrophoresis of sweat proteins should be done. The result of this test will be positive reaction for sweat specific protein and negative reaction for known sweat samples. So this test is actually confirmatory test for sweat. The significance of this test is that this is more specific to sweat and differentiate from other fluids. Okay, because the dermicidin, which is a unique peptide that is only present in sweat, is tested in this test. So DNA profiling of sweat is also possible. Sweat actually itself is watery, but it can contain like shed epithelial cells from skin, as I had told you in the previous videos. So DNA can be extracted from those cells, right? So procedure of this uh, test is that swab the sweat stained area and extract the DNA, then STR analysis is to be done. The result of this test is that it produces individual's DNA profile. It actually provides a unique DNA profile of the person, right? So person identification is possible. What is the significance of this test? This is actually extremely used in cases where suspect touches the surface or left sweat on any clothing. Okay, now we are going to study about vaginal secretion. This is also a very important body fluid, which is often found in sexual assault cases. So the principle of this test is that vaginal fluid actually mildly acidic in nature, like almost 4 to 5 pH. Mildly acidic it is. The procedure of this test is that um, we just have to like apply pH indicator onto the suspected stain. The result will be acidic range color change, like from dark red to almost yellow, right? This is the range of acidic substance. So this is actually not so much specific. but this can support the detection, right? Next test is acid phosphate test, which is also a percentage test. The principle of this test is that vaginal secretion also contain acid phosphatase enzyme. Okay. So as I have told you earlier, in semen also there is acid phosphate test. So this can say that acid phosphate test is actually not at all specific at all. This can be present in semen, this can be present in vaginal secretion and any other body fluid also. So the procedure of this test is similar to that we have seen in the semen sample. We have to just add alpha naphthalene phosphate and then fast blue B to the extract of vaginal secretion, and then we can see a purple color. Like purple color might be weaker compared to the semen. Okay. The significance of this test is that can give false positive test due to overlap with semen and other fluids. Now, confirmatory test for this vaginal secretion is that microscopic examination. Like vaginal epithelium actually has glycogenated epithelial cells, which are very distinctive, which are very unique to vaginal fluid. So the procedure of this test is that we have to just stain the sample with Lugol's iodine reagent, and the result will be glycogenated cells. Stain them down. Okay. So yes, the significance of this test is that indicate this test actually indicates vaginal origin. Like it is actually useful in sexual assault cases where vaginal fluid should be detected and confirmed, right? So the next test is RSID vaginal test, confirmatory test. So the principle of this test is that immunochromatographic test for human vaginal specific proteins is the basic principle of this test. The procedure of this test is that we have to just apply the extract, the vaginal extract, the vaginal secretion to the RSID test strip. Like if it produces colored band, then that confirms the presence of vaginal secretion. The significance of this test is that it is very highly specific, like for human only. So human strains can be correctly confirmed by this test. So this is actually used in modern forensic laboratories very well. So the next one is DNA profiling. This is also a confirmatory test, as we know. Vaginal secretion also has epithelial cells in it with nuclei. Okay, so the DNA can be extracted from those cells because it has nuclei in it, and then we can actually perform profiling. So the results will be produces unique genetic profile of that person, right? So we can actually link the suspect and victim to a crime scene, to a sexual assault. So that's all for today. Thank you, actually. And if you have liked this video, please like, share, and subscribe to Think Sakshi. In the next video, we will be learning more about the seminal stain and the sexual assault cases. So stay tuned. Till then, have a great time.